many different products that are already being targeted towards companion animals that are kefir based, which is a fermented dairy beverage, or even yogurt based, or I'm sure you guys have talked about on the podcast, but pet foods that have probiotics or postbiotics, things that are technically supplemented into the diet to have, other effects on the dog's or cat's health.
Well, welcome to the Pet Food Science Podcast. Of course, we're here with experts in the pet food industry and nutrition of pet foods. I'm very pleased to be with Breanna Metras today. We're going to talk about some specific new ingredients and sort of perhaps even how that might influence pet nutrition. And then I'd be very interested in talking with Dr. Metras about her career path and how she got to where she is today. So if you would start that, Dr. Metras, why don't you just tell us where you are and how you got to that place?
Yeah. So hello, everybody. My name's Breanna Metras. I have my PhD in getting my registered dietitian. Right now, I am at the University of Illinois in Urbana-Champaign, although I originally hail from Boston, Massachusetts, which is where I grew up and where I did my undergrad. And so I'm here now finishing my degrees, and, I'm looking forward to what the future holds. I'm currently doing my dietetic internship, which is part of the requirements of becoming a practicing dietitian. And so I'm juggling a lot at the moment, but it's very fun. I'm at the end of my schooling, so it's been a nice kind of closure period for me.
Leading pet food manufacturers, renderers, and ingredient suppliers recognize that Kemin is assurance. Every day, they deliver specialized expertise, innovative products, and unrivaled support through the pet food and rendering value chain. From oxidation control and food safety to palatability and nutrition, all the way through a suite of tailored services that allow you to feel supported from start to finish to ensure you're getting the most from Kemin ingredients. That's why every step of the way, Kemin Nutrissurance is your partner in pet food and rendering ingredients.
What made you get interested in this? How did you decide that this was a career for you?
Yeah, so when I did my undergraduate degree in biochemistry, and I also worked as a server in Boston restaurants and fine dining. So I have a very strong passion for food and food science and kind of how the different parts and components of food really affect us inside and out. And so when I was taking my biochemistry courses, as soon as we started learning about vitamins and minerals, I was like, oh, this seems a little bit more applicable. And then I did a lot of research in organic chemistry labs. And that was a huge disconnect for me because I really wanted to be working with people or making an impact on other people or dogs and cats and their health. And I really didn't feel like I could do that from the bench in the lab. So I decided to pursue my PhD in nutritional sciences at the University of Illinois in Urbana-Champaign. And Dr. Kelly Swanson is my advisor, and he does a lot of companion animal nutrition research. But he also, in addition, does have a small section of his research focused on applications in mouse models, as well as in vitro experiments that could be translated to, other clinical models. And so for me, that was a perfect fit because I got to apply my nutrition passion to this field, but also it gave me a chance to kind of grow my knowledge base from not just humans, but in addition to cats and dogs and companion animals. So it's been a great experience for me to, try different things and get to understand how the different fields work, but most importantly, how they connect with each other, because there's a lot more similarities than we realize in terms of marketing and consumers and different products. So it doesn't really matter where you start, if it's humans or animals, it's kind of what you learn along the way.
Well, that sounds great. It is nutrition and sort of, obviously there's a tremendous overlap in the monogastric nutrition area between human nutrition and dog and cat nutrition. Although I always sort of smile and say, yeah, but cats are still very unique because cats
Yes.
are probably a little further away in that continuum.
Mm-hmm.
You worked, and I know we were chatting even before we started this, you were working with fermented foods. And,
Yes.
That's a lot of people maybe sort of familiar with fermented foods. I think of kimchi pretty quickly, or some other foods, but in pet nutrition, maybe not so common. So tell us about that. What did you see of interest, and the background first, and then we'll maybe dive a little bit into the results, because I think that's the kind of key of this podcast for me is, well, let's talk about this new area, if you will. I personally have never put a fermented food in a pet fermented food ingredient in a pet food. So let's talk about it.
Yeah, for sure. So this research kind of was born out of my impatience of starting a research project with Dr. Swanson. I really wanted to get into the lab and do a mouse study, and he's like, we don't have time at this moment. So instead, I went ahead and we did some evaluations of kefir products. And first we started with human products, but due to our lab's heavy hand in nutrition science and Kelly's, well known among industry as well, he had been aware of many different products that are already being targeted towards companion animals that are kefir based, which is a fermented dairy beverage, or even yogurt based, or I'm sure you guys have talked about on the podcast, but pet foods that have probiotics or postbiotics, things that are technically supplemented into the diet to have, other effects on the dog's or cat's health. And so we really started to realize pretty quickly that it's a wild, wild west of A, companion animal supplements, but B, the fermented food category and that whole new age era of supplements. And so upon, further investigation, we realized that there's not a lot of regulation, which is also true with human products of this nature, but that the claims of health were very grandiose in the pet food area, more so than I think we see in humans, because these products are able to utilize their whole website platform, their social media, in addition to their bottle. And consumers are really eager, to give their pets the best nutrition possible. And if this new ingredient or new fermented food is the key to that, people will shell out and people will purchase those products. And so it was really important for us to take a minute and survey the field of what is out there for fermented foods for pets. You don't see a lot that is marketed outside of dairy products, because dairy is a really common fermented food for many animals, or mostly humans. But, my cat will eat the yogurt, he will eat the kimchi. No, I'm sorry, not the kimchi. His name is Kimchi, by the way, my cat. Yeah, but he will eat the kefir, I meant to say. So, they're not opposed to it. They'll eat anything that their owners, will provide them as a quote treat. And what it actually does to their gastrointestinal system, we're still figuring that out as time goes. But if we're starting to see, benefits in terms of bioavailability, micronutrient density, or, microbial effects from humans, why not say that could also happen potentially in animals who are consuming these?
Well, that's certainly possible, isn't it? So there's a lot here to discuss. We, as I look at what you said, you talked about bioavailability, you talked about these prebiotics and postbiotics all sort of wrapped up in this fermented ingredient. So let's break that down first. Let's remind us again what you mean when you say prebiotic and postbiotic, and how that might be influenced by this fermented ingredient.
Yeah. So prebiotics are a type of usually indigestible fiber that is to serve a benefit to the host when consumed. These prebiotics are usually fuel for microbes in our body due to the lack of enzymes humans have, or animals as well, to break down this fiber. And so the term prebiotic is essentially meaning like fuel for the microbes in your body that can be beneficial to you. And then postbiotics can be, I can't remember the exact definition, but it's basically an inactivated bacteria. And so even though they're not active, the dead cell bodies and the dead bacteria that are remaining in the product can actually exert an effect as well. And so how those actually do it, we're still looking into it.
And I think postbiotics, at least as I use it too, also includes not only those bacterial components, but the sort of milieu that comes out of bacteria. So you could look at something like butyric acid as a postbiotic, right? That the bacteria made has value to the pet. or the person. As we've discussed, these are common. Well, how do you sort of know what's good? I mean, how would you evaluate this to know if there really is a benefit?
So we can evaluate the product in terms of the product's accuracy in itself of being, efficacious and truthful and having integrity in terms of, stating what they have and what's actually in the product. But then we can also look at what's beneficial in terms of the physiological outcomes that we have on the animals. And so a lot of research, at least for animal studies, I'm sure, as you know, are done with like lab animals that are typically very healthy. And so when we see benefits of fermented foods in people, usually there's some kind of pre-existing health condition that is improved. Maybe that's your A1C level for diabetes or hypertension levels or blood pressure, blood lipids. And so in order to test these, also test these measures in dogs or cats, we would need a population that is not exactly physiologically perfect. And so when we do our research with our research dogs, they're really well taken care of and they're really healthy. So measuring physiological differences can be tricky, but we can still look at how modulation of their gut microbiome via fecal collection is happening. So it still gives us an insight as to what is changing, what's being affected, what microbes are thriving or not, and how that can be extrapolated into other scenarios for, the average pet consumer.
Well, that is fascinating. I think we should at least contemplate it, maybe chat about it a bit. Was what you're saying is we feed to the normal pet, right? So we're feeding a population of pets. We try and hit that optimum mean, really. And yet a lot of the action is at the extremes. So that perhaps that extremes and testing that extreme gets really hard because say you have even a population of normal pets out there at the tail, that might be 1-2% of the population. But if you're a pet nutritionist, you may be feeding a few million pets by the food. Certainly those tails become pretty significant. So what I heard you say is you're really kind of, wishing for, or you're measuring markers that might be indicative of that influence, and that marker was a change in the microbiota. Can you talk about that a little more? How do you measure that, and what might you be looking for?
Yeah. So looking at the microbiota is a way I like to describe it as kind of like a window into that person's health or that pet's health. And, it's just a viewpoint. So it's not really indicating, a certain health marker or disease or optimization. It's just like a single snapshot of that moment of time of what their gut microbiome looks like. And so in terms of, health, we do look for specific bacterial species or genera that are associated with producing beneficial byproducts such as short-chain fatty acids. So these are a type of small compound that are volatile, and they actually serve as fuel sources for other microbes coexisting in the gut microbiome. So if we see some animals' microbiome, we see their data, and they tend to have more short chain fatty acid producing bacteria. For example, maybe butyrate, which is a really common but highly beneficial short chain fatty acid. Then we can see that, okay, this animal has a balanced microbiome where we don't have certain harmful species overpowering beneficial ones, and we still have a balance. And so looking at that is really important, but also looking at the predominance of different phyla. So within different in the microbiome, we have different phyla of bacteria. Most common in dogs is Fusobacterium, but we also see firmicutes, proteobacteria, and bacteriota. And so the interchange of those larger fractions of microbe groups kind of also gives us a picture of how our intervention is really affecting their health in terms of that microbiota snapshot.
No, I was just thinking, I wonder if we could talk about it a little bit, that, Dr. Swanson and others have been working around this, how do we describe a healthy microbiota? Because it seems like you can have pets with very different microbiota and be quite healthy, or if anything, the consistency becomes in the unhealthy ones. They begin to really look alike, and the healthy ones can look really quite a bit different. Is that your perspective, or how
Yeah.
do you handle that?
Yeah, and that's a great question because that's something that the FDA is also wrestling with at this moment. How are you going to classify what is a healthy, phenotype or healthy class of a microbiota when every individual, has extremely different than their neighbor due to their diet, genetics, environment, and the same goes for pets. And so kind of being able to classify certain bacteria, as being beneficial producers of short chain fatty acids is one way to look at it. But then again, if in another context, for example, if a dog has like IBS or IBD and they're highly inflammatory and they're gastrointestinal, those beneficial bacteria are going to be much lower in terms of content. And so that's one way to look at it. But in terms of a like diagnosing perspective, the microbiome is kind of the after effect. So we see what's happening when we look at the microbiome, but we can't use that information as a tool to decide if that animal is healthy or not. It's more of an indicator of where they're at the moment.
I understand. That makes sense to me. I do have to smile at once in a while you can really tell if they're not healthy. If Heliobacter is 50% of everything you recovered, then it's not going to work well for that pet.
Absolutely. Yeah. Those extremes are really telling.
Yes. What do you think? Sometimes I wonder what your opinion is there kind of like gatekeeper, small numbers, but wildly important bacteria, so that those bacteria really influence a whole spectrum of bacteria. But if you were doing them as a percentage, they're not going to show up very high. What do you think about that?
Yeah, I think that's possible in terms of like smaller existing microbes that maybe have a certain fitness to digest specific compounds or nutrients or phytochemicals. So that is a potential. I personally am not familiar with specific smally existing species, but as technology gets better, we could really identify those smaller species by not just their species, but their strain, because the strain is where the genetic fitness is kept. So something can be, Lactobacillus acidophilus as a species, but their strain of that different acidophilus is what really determines whether or not that microbe has the potential to break down a vitamin or a phytochemical, or in this case, maybe digest an element of a postbiotic. Yeah. And so once we get better technology in terms of identifying the genetic fitness of these microbes that are existing in a small quantity, I think then we can try to optimize them to pair them with a specific diet or a specific nutrient supplement that they're, programmed or optimized to work with. But at the moment, from my at least clinical experience, those very small existing microbes will have, depending on the time, but mostly minor effects, just because it's a huge competition. It's a big old party in there. And so when you have such small amounts of one thing compared to the bigger groups of, the commensal bacteria, it can be kind of hard for them to make a difference, but it's not impossible. That's for sure.
Interesting. Well, let's dive a little bit into your research. Now, you've published a couple of papers. Just tell us what you were studying and maybe what we should take away from those results.
Yeah. So my research, I'm initially studying the fermented beverage kefir. And first we looked at the product efficacy of kefir items on the market that are geared for companion animals. And so we analyzed, I think, 6 different kefir products that were targeted for dogs based on their label claims of microbial density. So how many live microbes will be in the product when you serve your dog, as well as the microbial ingredients. So what intentional microbes were added to this product that you are ideally serving your dog. And so our research really looked into, we extracted the DNA from these products to better understand what bacteria are there. And we use this technology called loop sequencing, and it's a high throughput long read 16S. So we read the whole genetic region for those bacteria to really understand who was there. And we also culture these products on agar that are beneficial for lactobacillus, as well as agar beneficial for yeast, so that we can really get an idea of who's in this product and who's still alive. And so most of our research found that on average, the dog products were not fully accurate. Most commonly in the microbial content, many things were overstated, but as well as we found a lot of species that were not disclosed on their label. Now, that is not at a fault of their own, because they might not have the technology the university has, to look into what is actually in these products. But moving forward in the fermented food space, retaining this integrity is really important for consumer trust. And we also found that a majority of the products do not have a lot of microbial viability at the point of consumption compared to what they claim. So many will say, oh, we have like 10 million or 1 billion live, microorganisms at consumption. When reality is, at least with the pet products, many of them were under 1 million, and some of them didn't even meet our threshold. So we really didn't get a lot to grow. Now, we, of course, had a control. We used Chobani yogurt, and, that met most of our, it passed all of our checks. So we are confident that our methods were okay, but, it definitely begs the question of, are the products okay? And, should you be spending so much money on it if they're not quite what they say? And to kind of further into see, are they beneficial for dogs or not, we did an intervention study. Did you want to ask any questions about the first part?
Yeah, if I could. So this is a study where, if I could summarize it, you were looking at a spectrum of products, and among other things, you were checking, are these microbes alive? And you checked them being alive by actually trying to grow them in culture, which that's not an easy thing, right? So that's why you said, well, we have a positive control out there, so we know at least those microbes can be grown.
Right.
Do you think, like, one of the problems I had back home, so long ago, I don't want to talk about it, when we started looking at feeding the lower gut, we had a tremendous inability to grow the spectrum of microbes that were there. And I know, certainly you do it better than we did in the 70s. But still, that's hard, right? So some are commensal, some are anaerobic, some do fine aerobic. And
right.
Can you talk a little bit about, why a consumer might believe that test was at least a reasonable representative of what was actually there and alive?
Right. Yeah. And so we really took steps to, optimize our conditions so that we could really cultivate what was present. And just to note, the microbes that are in kefir as a dairy product will vastly be different than those collected from a gut sample or from a fecal sample. So culturing them, I wouldn't say it's quite as challenging as culturing from, an actual fecal sample. Just due to the fact that, they're predominantly lactobacillus, which is a very easy to grow in specific nutrients. We use manna rose sharp agar, which is, really common for lactobacillus. So we had confidence that we were going to grow the predominant species that were present. And yes, we did end up having to grow them in anaerobic conditions, so without oxygen, in aerobic conditions, due to the fact that there's a lot of preferences among those bacteria. In addition to that, we grew them at 2 different temperatures. I believe we did 30 degrees and 37 degrees Celsius to try to optimize and really capture different ends of the spectrum from like where their happy place was for, growing over those 2 days. So we really did try, and we also serially diluted the products where they're so highly condensed in terms of microbes. You can't just plate it as is. You're going to have a whole bunch of happy consuming microbes. So we had to serially dilute them by, I think we ended up keeping the 10 to the 5 and 10 to the 7 reductions just because there's such a high concentration.
And that's why you had the million cutoff at 10 to the 6. If you didn't have 10 to the 6, then that reduction is just going to blow it away. Got it. Well, that sounds interesting. And so you feel comfortable that if you evaluated them, at least the lactobacillus, you could grow them, and you showed that you could grow them by the positive control. So that just makes sense to me. So then you went the next step, right? So it's nice to have an agar with lactobacillus growing, but you as a consumer or as an individual, you kind of go, well, what does it matter to the pet? And you actually fed these to the pet. So how did you do that, and what were your results?
Mm-hmm. Yeah, and So lactobacillus is an interesting microbe because it's very common, like I said, in dairy products, but it's a little bit less common within our own gut as gut commensals. They do exist, but they're not the predominant species at all. So when people are consuming probiotic supplements or fermented food supplements, a lot of the predominant species in those items are kind of like tourists. They're coming through our body, they're visiting, they're saying hello, and then they're leaving. They don't really stick around very much. So the idea is to consume these products regularly to kind of see that effect by repeated exposure. So when we did our dog study, we had 12 healthy dogs. We did a 3x3 Latin square design. So we had 3 different treatment interventions. We used the commercial kefir, which is one of the ones that we analyzed in the study. That is actually produced in, I think, Bloomsburg, Indiana. And then we also made a freshly brewed grain kefir. So traditionally, kefir is brewed from the inoculation of these bacterial grains. They're just like a coagulation of bacterial species that look like a grain. And you inoculate the milk with these grains and let them sit on the counter for 24 hours. The resulting product will be a very effervescent, a little sour, a little tart, but really refreshing beverage. And so there's a lot of discrepancy in the world of research, whether or not it's a lot of intervention studies are actually using the grain kefir or the commercial kefir, like what we tested, because their microbial profiles are extremely different. Their food matrices are relatively similar, but in terms of nutrients, exopolysaccharides, byproducts, proteins, they're vastly different. So their beneficial effects could also be contrasted. So we gave these dogs one of those 2 products. In addition, we did a milk control, which we treated with lactase because dogs don't really like lactose very much.
Right.
So we wanted to cover our ground. We didn't want to clean up any messes.
I understand.
Yeah. So, and the way I administered it is we gave the dogs the intervention twice a day for 28 days in each period. So there's 3 periods. And basically I weighed out their food like we do normally for each dog. And I would pour 30 milligrams of that beverage on top of their food. And we did that for breakfast and dinner. So they were consuming it twice a day for 28 days straight before we switched treatments. And so that was how it went. And they really enjoyed it. The dogs were not picky. They loved the milk. They loved both kefirs. So that was a huge relief. That would have been a tough study if they didn't want to eat that.
Yeah.
But there was no complaints. There was all clean bowls. So that was a pretty successful intervention in that element.
And what were your results then? What did you measure as a response variable, and how did those change if they changed?
So we measured blood metabolites, so serum markers in the blood, such as lipids, glucose, different protein presence or not, such as like C-reactive protein. We also measured immunoglobulin A, which is a marker of mucosal integrity as well as immune functioning. So we're really hoping that, immunoglobulin A would have been affected because that would at least show some kind of immune optimization with these already healthy dogs. We also measured short chain fatty acid production from their fresh fecal samples that we did on the last day of the study. And we looked at their gut microbiota as well. So there are several different markers of that in addition to, total digestibility of the diet. But we expect kefir to be very digestible because it's, mostly water, high protein, maybe some sugars. And so that wasn't a big worry. But so as our results, we didn't see any physiological differences among the treatment groups. So that was a little surprising, but also, like I said previously in our conversation, dogs are healthy. So, seeing improvements in their inflammation or blood lipids or glucose tolerance or inflammatory markers, they're already in a pretty good normal range. So to see those be optimized from a simple product intervention was unlikely. So unfortunately, we didn't see any difference with that. But we did see a couple of significant differences in the gut microbiome space. Most notably was an increase with the traditional kefir intervention in lactococcus. And so Lactococcus lactis is a bacteria that's predominant in the commercial kefir product, but also predominant in the traditional product. And so the fact that it might have colonized by giving it to the dogs repeatedly through the traditional kefir, but not the commercial, was pretty interesting to us because, like I said previously, many probiotic fermented food interventions do not result in colonization. They're kind of passing through, exerting their effects, and leaving the system. And so we saw about 0.39% presence of, the lactococcus, which is not insignificant by any means. We do have, like we talked about, lots of bacterial species and some at very tiny levels, but something that's, over 0.1% of the presence of a whole is something that we definitely pay attention to as researchers. So that was the major output for that. And so although it might seem insignificant in terms of applying that to a dog who is experiencing inflammatory diseases or symptoms, having a increase in, maybe an anti-inflammatory bacteria like lactococcus could be, interesting or potentially something that could help their symptoms down the road. But we definitely need more research to see that.
So you saw in those healthy pets, you saw a benefit in the shifting makeup of the microbiota, but the more research is really needed to say, yes, but in that unhealthy pet, maybe out there in the fringes where we were talking before, that would turn out to be a whole pet benefit. I wondered if you said something that intrigued me. I wondered if you could dive a little deeper into it. You talked about this, was it anti-inflammatory sort of response from a microbe? I mean, how does that work?
Yeah. So microbes will produce something called a cytokine, or it's basically a chemical signal that kind of will communicate with the surrounding immune system to give a little bit of a gauge of where that particular area of the body is at. So when anti-inflammatory or signals that, indicate we don't have any reactive oxygen species or oxidative stress, that, we're trying to tamper down an effect of inflammation, that is a sign. And that's a sign that, there's something going on that is more active rather than passive with this intervention or with this species. And not to demonize inflammation in any way. Inflammation is happening right now in our bodies. It's always going to be there. It's a part of the balance of our homeostasis. And so sometimes we don't want an entirely anti-inflammatory response as a whole. But when you're feeding a dog or something that is already experiencing symptoms of inflammation, having some agent that might reduce that and create that balance again could be helpful.
ProAmpac, your companion in pet packaging. Visit pets.proampac.com to explore our innovative sustainable solutions, such as our QuadFlex recyclable flat bottom bags, ProDura polywoven bags, ProEvo recyclable paper bags with grease resistance, and our proactive recyclable film and pouches that run at optimum speeds on your filling equipment. Elevate your wet food, treats, and kibble brand by utilizing packaging that safeguards and preserves product freshness. Trust ProAmpac for packaging that cares for your pets and the planet. pets.proampac.com.
I understand. So inflammation in balance is what we want. A zero inflammation would probably be death. I always use
we can't do anything.
Yeah. I always use the fire in the fireplace. We want that. We get a benefit from that. It escapes from that, and that's when out of control is not good. I wonder. Then would you talk about, or did you measure? I can't remember. Did you measure cytokines in your response, or would that be like another step?
Yeah, in this particular study we didn't, but it would be a great opportunity to take this experimental design and look into that.
Yes.
Like I said, due to the fact that we're working with healthy dogs, we're not going to see a lot of improvements, but in terms of overall production or reduction in inflammatory ones, that would be great. There's been research with humans in terms of consuming fermented foods where we do see a whole cohort shift of decline in inflammatory cytokines. So it'd be very interesting to see if, this pattern is also existing in other animals or other models with specifically kefir.
Yeah, that would be interesting, wouldn't it? A good next step. Any research project or area always sort of shows the light where we might go next. I liked your illustration of the window. I've used that myself, where you a window opens, you look through it, but you see only part, and so
Yeah.
you want to observe more and understand more, and certainly that indication of what was happening with the cytokines, either circulating or upon stimulus, right? So there's some work that could be done where you isolate the immune cells and look at how they respond to a LPS or some stimulation that would perhaps
Yeah.
be affected by the fermented foods.
Yeah, I think we were really hoping to see some kind of effect in IgA or immunoglobulin A to kind of justify further cytokine research or, immune cell research. So yeah, we weren't able to pursue it in that study, but yeah, in the future, that would be a great place to look.
Sure. Now, well, I wonder, as you look back at your time, and I always ask this question to people, I wonder if you'd kind of think through it with me. You've been involved with a number of teams. You've been in a very successful research environment there. What would you say were the basis of that? Can you draw any kind of thoughts that would say, this is what I perceive as a really, really valuable thing for pet food scientists to be involved in a career? What would you be looking for?
Yeah, I think that, well, first of all, the University of Illinois, we have excellent animal research here. We also have a vet school that, watches over our dogs. So that really assisted a lot of our research and, making sure that everything that we were not doing was being taken care of well. So we really only had to worry about our intervention. But also, I feel like mentorship is really important in terms of where you do choose to pursue your education. And so Dr. Kelly Swanson is a great mentor. He's very positive, interested, enthusiastic, but also has his finger on the press of like what's going on in the pet nutrition world at the moment. So, that's been a really beneficial element to kind of have as part of this career. But also having a supportive lab, people that are similarly motivated, to do their own work, but also help move the team forward, has been extremely invaluable too.
That makes a lot of sense. It's fun, isn't it, when you see the sum of those parts coming together and being so much greater than just the individuals would have been. That's great success in a lot of ways.
Yeah.
So what's the future look like? What do you want to do as you walk out of this the steamed ivory tower?
Yeah, that's a great question. So that's something I'm working on right now. Originally I have a lot of experience in coursework, and I'm a certified in policy in terms of pet food, but also human nutrition. And so working on some form of dietary regulations or guidelines would be best case scenario. But I've recently realized that you can't actually apply for a government position until after you graduate. And so since I have a graduation coming up, that's something that I might have to table for the moment. So I'm also considering looking at a couple postdoc opportunities. I'm from Boston, and, there's a great opportunity at one of the schools there. I don't want to jinx myself, but, that are doing like really good work in terms of policy, but also nutrition interventions for low income and obese people in the city of Boston. So that would be great. But, right now I also do a lot of work with probiotics and, for the fermented food market. And so working for potentially a yogurt company or some kind of industry company that is working to develop either better or more precise fermented foods or precision fermentation, that would also be a great utilization of my education. But because I get the RD, PhD, I definitely have strong inclinations of counseling privately as well. So I really love that people element that you get from, just seeing people like we're doing right now. That like really feeds me with energy. And like, that's what I love. I live off of that, so having a desk job is one thing, but working with people day in, day out would be the best case scenario. Wow. So definitely not working remotely.
Well, I understand. So it's good to know that about yourself too, because some things that might seem really valuable to another wouldn't fit what you've just described. So best of luck with that. I wondered if you wanted to talk a little bit about those. I'm back into these fermented ingredients that might be, and I'm not asking for individual names of ingredients, but if I was a consumer and I sort of looked at your research and said, and maybe I find success self consuming these fermented foods, and I'd like to impart that to my pet, what might they look for? And again, not names, but what might they look for to say, this is one I want to buy?
Yeah, so I feel like, first of all, if you're going to buy some kind of fermented food, it better be refrigerated. There's a lot of times where you're going to see them shelf stable, or, they've just been sitting in a bottle and you just have to open it and you're ready to go. Those unfortunately are not the best purchases, just because we don't know in terms of research that that's a great storage method. From what we've seen, it's not. And same with dehydration or compression of supplements. When you are compressing a microbe, you're really affecting its composition into a smaller space than it really needs to exist. And so looking at pills or supplements in that form, I would also not be the first to recommend that. I would definitely go for something that's refrigerated, fresh, but also ones that are not trying to claim too much on their label. If they say, we're going to cure this dog's heart disease and fix its coat health, and, all these grandiose claims that, can't really be proven, for me, that's a big red flag. I'm like, okay, they need to stay in their lane. And the fact that they're not, kind of shows me where their priorities might lie. I really enjoy products that are more research based and research developed, because they can be a lot more accurate and, less exaggeratory. They don't exaggerate as much with what these effects can actually be. But research will come along, and I hope one day there will be regulated health claims for, for example, pet supplements, like we do see for humans. Not enough in the supplement industry for humans as well, but there's always room for improvement and growth. And I hope that, with my work and hopefully my career, we can, work on getting that more clarified for the consumer.
Well, that makes a lot of sense. I like those points. That sure rings true to me. What if, as a consumer, though, I followed that, I buy the best one I can, and now I'm feeding my pet? How would I know that this is a benefit?
Yeah. That's it's a great question. It depends on what you're looking for. I feel like right now, a lot of these benefits are really minute, unless you're getting regular blood work done for your pet and you're sequencing their microbiome every couple weeks. Other than that, it's kind of hard to get real heavy takeaways. So okay, this is actually making a difference. In terms of there have been, in vitro studies that, some of these proteins in fermented foods that, are specifically coming from, for example, traditional kefir, might improve certain elements of, skin health, or might improve elements of insulin resistance or sensitivity. And so those are the kind of things that you could test, maybe by visually looking at the skin or the coat of that dog, but identifying that is the reason, just the probiotic is very difficult to do.
Sure. And as a consumer, that you always have an N of one and it's an open study. It's not the same thing as you've done there. But, I can see looking at that and saying, okay, maybe even with today's prices, it's not so unusual to think I could do a microbiota of a fecal and sort of run that every once in a while. And if skin and coat started looking better, and I would think stool quality would be something one would hope for, that would be improved. So that all kind of makes sense.
Mm-hmm.
Well, I've enjoyed our discussion. I wonder if you just had any other thoughts about, say someone was interested in, and I know you had this great experience, but just in general in the pet food area, and perhaps pursuing that as pet food science. What would you tell that individual?
Yeah, I would definitely tell them to figure out what they're passionate about. Me particularly, I've always really enjoyed fermented foods, and, finding a way to kind of merge that passion with an impact, or, finding how that could connect to a consumer or a companion animal. has really been the peanut butter and jelly, because I'm really passionate about one thing, and to help other people or animals in a research facet is the best way. So, really identifying what gets you going, what really, lights you up, and then how you can bring that to the pet food industry would be key, in my opinion. And, there's many different ways to do that, but through research and then getting a job, you can be on an interdisciplinary team or work with a team of pet nutrition scientists, to really kind of get your ideas off the ground, or even, provide that valuable insight to that interest that you do have.
No, thank you. I think that makes sense and would be good advice for someone who is interested in pursuing this career.
Mm-hmm.
Well, again, thank you for being with us. I think this is a fascinating area, one where there's a lot of opportunity as we do investigate fermented foods for ourselves and for our pets. I'll look forward to that developing, and perhaps your leadership as you bring that along. And always difficult with the regulatory aspect. We have DSHEA, right?
Definitely.
And so that's out there, and the pet space maybe aligns or doesn't align with that. I'm not even sure. But
yeah, I don't think so. Not the way we hope. Yes, much to come for sure.
Thank you for your time, and we'll look forward to your success in the future.
Great. Thank you so much for having me today. I really appreciated the conversation.